because I know some of the people here probably know a lot about grid cells. Jose, maybe you probably know more than I do. I don't know. But my knowledge is a bit old. I haven't really read any papers about it in the last few years, so maybe 3 years or so. So I'm not up to date on the latest. But we were into it pretty heavily for a while back in the days, and of course it's not clear how much of this is relevant or necessary to know for Monty and the Thousand Brains project, because we're not going to emulate or model grid cells, at least that's not the plan at the moment. So I put down some basic things about grid cells, what we know about them, what we don't know about them, and how we thought about them for Thousand Brains theory. And I guess in some of it's going to be kind of simple for those of you who know this already, and some of it could be really hard if you've not really been exposed to this. I remember it took me a few months of real deep confusion before I really got it. It was like I kept getting confused over and over and over again. Like finally it sunk in, like, oh, that's how these things work. So if you're at all like me, it may not settle in on the first couple of exposures. But with that, I thought I could just jump right into it, and unless someone wants to say anything before I get going.
Yeah, I just agree also that it took me a while for it to sink in.
Yeah, it's kind of odd. It's really odd. So I'll just get going. I'm going to share my screen, and then I'm going to show you a PowerPoint presentation I have here. And not a lot of prep went into this, so really the best to make this can we get rid of this stupid thing in here? Make this interactive if we can. Hopefully you see that. Do you see that list of text?
Yes.
Okay. So this is just talking points I wrote down. I have images that go along with these, which I'll show you in a second. Let's just start at the top of this list. The idea basically is that the brain has to have some kind of reference frames for various things. It needs to know where things are in the world. And we deduced that each cortical column also had to have some sort of reference frame. And this is it's a pretty solid deduction, especially for somatosensory, for touch, where that's the most obvious place. When you touch something, if you're going to have an expectation what you're going to feel, then the brain has to know where the finger is, or the patch of skin is, or whatever you're touching, and it has to know where it is relative to something in the world, like an object you're touching. And as you move your finger, it makes new predictions. Therefore, it has to keep track of where your finger is. It can't know. It has to have neurons to tell it this. So that's a pretty solid deduction. And then it was kind of hard to imagine initially how neurons could create reference frames. But then we said, hey, there's these things called the grid cells in the entorhinal cortex, which is related to the neocortex. It's if you follow the end of the neocortex to the very end, it starts getting wonky and it turns into entorhinal cortex and hippocampus and things like that. And so there's and a lot was known about grid cells. We didn't know a lot about them in the beginning. So we said, hey, obviously the evolution has discovered a way of doing a kind of reference frame using these things called grid cells. So and it turns out they're pretty complicated. So we guessed that that the cortex uses grid cells too. And now we know that there are many places in the cortex that do have grid cells. And we made an early mistake, the third bullet here. We didn't really deeply understand this one either, but columns not only need to know location of a sensor, they need to know the orientation of the sensor patch. So it's like you can touch something with your finger, but you can rotate your finger on that point, and the input coming from the finger changes, and the cortex needs to know not only where the finger is, but its orientation to the thing it's touching. And these are similar to grid cells and hedge-Rexin cells, which you see in the hippocampal complex. All right. So we said, okay, this basically, we think that cortical columns are doing something relatively similar to what's going on in these other parts of the brain. And I'll point out here, the last point here is that grid cells have been heavily studied. There's a huge amount of literature on them. A lot is known about them, but there are many things that are still mysterious. There's a lot of details about them which are argued about, no one really understands. Or if they do, I haven't read about them. Maybe they figured it all out in the last few years. I don't know. I doubt it. But there's still quite a lot of confusion about exactly how these things come about and how they work and so on. And I'll talk about some of those. some of the things we don't know. So then now I'm going to just go into grid cell basics, and then later I'll come back to talk about some of the things we don't understand. If there's any questions,
Maybe-
just shout out.
Maybe one other thing, at least I found useful or interesting to think about is, I guess, how like the connection between evolution and how the entorhinal and hippocampal complex likely predates the neocortex and kind of maybe was a blueprint for the neocortex. And like you can kind of imagine how a more primitive animal, just needs to know about basically have a reference frame for environments. Like that's the most important thing to first have a reference frame for is where you're in an environment and how you move around it. But then that kind of idea of having a reference frame as a way of structuring information is kind of a powerful, more general thing. And then what was kind of this complex, specialized kind of part of the brain for mapping environments could then potentially be kind of turned into a more compact, more general structure, which is cortical columns, and then kind of repeated. I mean,
Right.
I appreciate that's
Yeah,
like speculation, but
it is
but it's just an
speculation.
interesting way to think about like how you go from just first
Yeah.
doing environments to like using the same concept for representing all information.
Right, and there's some other things that really suggest it. So try to imagine now the cortex is this sheet of cells. We say it's 6 layers. Just pretend it's 6 layers. When it gets, as I said, when it gets to the sort of the end of its sheet, it turns into it becomes less structured, less uniform. And one of the first things it becomes is it literally follow the sheet and it becomes the entorhinal cortex. There's other things first, and one can argue that the entorhinal cortex is, let's say, roughly 3 layers thick. And then it continues on and it folds over on top of itself. And the hippocampus is now on top of, it's like folded like a sheet of paper. And now you have like 3 layers of the hippocampus on top of the 3 layers of the entorhinal cortex. And so this is a very rough approximation, but it's kind of true. And so the idea there is, one of the ideas that we didn't come up with, but it seems like it's a reasonable idea, is that the 3 layers of the hippocampus on top of the 3 layers of the entorhinal cortex, then at one point in past history became the 6 layers of the cortex. And so there's some analogies between the place cells in the hippocampus being the upper layers of the cortical column and the entorhinal cells in the entorhinal cortex being the lower layers. And so I've always got that in the back of my mind. So not only did evolution discover this thing, but physically there's a suggestion that the cortex became the fusion of these 2 3-layer structures that are lined on top of one another.
Are you going to mention place cells as well?
Yeah, I will in a second. So why don't I just get into that and then we'll do it. So let's just start at the upper left hand of this picture. The first sort of cell in this collection of cells that was discovered was the place cell. And the discovery of the place cells led to a Nobel Prize by the O'Keeffes. This picture on the upper left shows a rat walking around in a square box. And the black lines show where the rat was walking. And the red dots are a particular cell in the hippocampus of the rat that fired. And it fires in the red dots of where it fired. And so this cell seems to fire at preference when the animal's in some location in this field. It's consistently so. And so this was the beginning of place cells. Now this is just one cell, but it seemed to say, hey, no matter which way, no matter which way the rat was facing, it doesn't matter. If he's in this area in the bottom right of this cage or whatever it is, a big box, the cell sort of fires and sort of marks a place. And so that was a key discovery, and a lot of research was done on that. And then later, another team discovered a similar cell in the entorhinal cortex called grid cells. And so in this case, the picture is a little bit fuzzy, I apologize for that, but the middle image there of the circular field, this is a big circular box the rat's running around in. And again, we're recording from a single cell here. It's the same idea. There's black lines saying where the rat was moving, and the red spots show where this particular cell fired. And it's consistent. It doesn't matter which way the rat moves around in the box. Whenever it gets into these locations, the cell fires. So it's sort of like some kind of a location signal. And I think this is just a heat map of showing where the cells were most active in those spots. And so you can see right now that this particular cell fires in a lot of different locations. This is just one cell showing where it fires as it moves around in these locations. And it turned out that this isn't a sort of a triangular or hexagonal pattern where this cell fires. And now, so that's pretty cool. Now, of course, this one cell doesn't tell you where the animal is. If you just looked at where this cell is firing, it could be in any one of these locations. So it's not a very good way of locating the cell, locating the animal, but that's what it is. Now, of course, there's lots of other grid cells, and they vary in a couple ways. First of all, there's a whole bunch of grid cells that share the same property of this one. So go down to the bottom right here, and you see the square that's labeled phase. So they're showing the activity of two cells here. This is a hypothetical, one that's green and one that's blue. And these cells share the same spacing in terms of their firing pattern, the same orientation in terms of their firing pattern, but they have different phases. So they fire at different locations and overlapping locations. And if you have you can find a whole set of grid cells that essentially tile the space all with the same orientation and scale. So whenever the animal's at any location in this environment or any environment, there's at least, there's a few grid cells that are firing and in the same grid cell module. And as the animal moves, the which cells fire change, but all the cells in the same module, as they call it, have the same orientation and scale. Now, there are other grid cell modules in the entorhinal cortex. If you move horizontally in some direction, you'll find a next grid cell module has a different scale. And so there'll be exactly the same thing we just saw before, but the cells will fire at different, different spacings. And so, and there's like, I forget how many there are, but there's somewhere between 5 and 10 of them or something like that that can easily identify like this. And so you get larger and larger and larger scales for the grid cell modules. In fact, here's a little interesting historical anecdote. The people who discovered the place cells, they didn't discover grid cells because, and O'Keefe wrote about this, because they were looking at cells that had a much larger scale, and therefore at the actual grid cells they were looking at, of course, they didn't know they were grid cells, but the cells they were recording from, the scale was so large that there was only one cell, the cell only fired one place in the environment, So a rat has grid cells that scale to cover very large areas, maybe 50 meters or something like that. And if you're looking at those, then you won't see that cell fire in multiple locations in a particular smaller environment like this circular arena. So that's an interesting thing. And then at any point in time, go ahead.
Do they basically have a map of where the larger scale grid cells are and where the smaller scale grid cells are, and that's how they can now say that where they recorded back then is?
Yeah, I guess so. Right. I think there's some. Yes, they do know. There's a particular direction you move across the entorhinal cortex where the modules get larger and larger, and this has been mapped out. I'll show you a picture of that in a second.
Okay.
Anyway, so by the way, just a little jumping ahead here. So you've got these multiple grid cell modules that increase in scale by, I think, like a factor of 1.4 each time. So it's a multiplicative factor, something like that, or maybe 1.7. I can't remember the number. Anyway, but it reminds me of this change in the scales in the regions in the cortex, like from V1 to V2 and V4. You have a similar sort of bump up in scale. And I've always wondered if it's the same process going on there, that literally the entorhinal cortex has got a set of grid cell, place cell pairings, and they go up and they bump up to the next size and it's the next size. It's very much like V1 and V2 and so on. Okay, any other questions on that?
So is the hypothesis that place cells are basically disambiguating the grid cell firings, or what's the interaction?
No, well, the basic hypothesis here is, and by the way, this is very heavily oriented towards my hypothesis. So there's been a lot of literature about this, but the general idea is that place cells are more sensory driven. So they're based on observations the animal makes. He sees something and says, "Oh, okay, I now know where I am," based on this visual input or tactile input or input from their whiskers or whatever. And grid cells are driven more by movement. And so these, to me, there's an analogy here between the upper layers in the cortical column, which are driven primarily by sensory input, and the lower layers, which are driven primarily by motor path integration. And that it kind of looks like that. So that's a very good hypothesis. That's the one I've always adhered to. There's a lot of detailed research about the correlations between grid cells and place cells, and I can't recall all of it. It's just tremendous.
Yeah, I think the invited speaker we had a couple of weeks ago, Haggai, he kind of looked at how they influence each other in a bidirectional way.
Right. So the simple way to think about it for us, I think, at least for me, is that the place cells are very similar to the layer 4 type of cells we have, and they're driven by sensory input, and they're paired with the location in the lower layers, which are more equivalent to grid cells. And so there's a bidirectional connection between those, and those connections do exist between the hippocampus and the hippocampal cortex. So it's a very reasonable hypothesis, I would say. There's also an orientation because these cells are spaced in like a triangular grid or hexagonal grid. There's a change in orientation. So when you go between grid cell modules, from one module to the other, not only do you have a change in scale, but you can also have a change in orientation. And I believe that the orientation only varies slightly, like suggested in this middle image. But they do change. And you'll see in a moment why that could be important. One thing to note is when an animal goes into an environment, if the animal recognizes the environment, then the grid cells, what they do is they call anchor. That is, once the animal knows what environment it's in, the grid cell that was active in one particular place before becomes active again. In fact, all the grid cell modules anchor as if they were in this place before. And the animal goes into a different environment that's not the same one, then the grid cells anchor differently. Meaning, maybe let's say there were 50 grid cells in a module that could be active. And so one is primarily active and the other couple are maybe not so active, and all the rest are not active. Well, which one is active at some point in time is a choice the system makes, and that's the anchoring. In a different environment, a different cell would become active at some point. In fact, you could have two circular environments like this, and if there's a picture on the wall, a rat will see these as different environments, and the grid cells will anchor differently but consistently between the two environments. So recognizing where the animal is, once the animal recognizes it ends, these grid cells lock in and anchor in the place they were before, and in some sense establishing the same reference frame that was established the previous time the animal was in this environment. And that is a big part of what it means to recognize where you are, is that once they go, oh, I know where I am, bingo, all the grid cells, and now you have this reference frame and everything can be predicted. But before then, your grid cells might have a different anchoring position, and you don't know where you are, or you think you're someplace else.
Is this something where like the place cells are really important in this anchoring process? Since
Well, clearly, this sensory input is very important in the anchoring process, right? Yeah. So it's very much like that. It's like a sensory input coming into layer 4 of a cortical column. That sort of suggests where you might be in the world, but it's not definitive, perhaps. But once you've had enough observations, then the layer 6 cells would anchor, and oh yeah, we know where we are. We're on the coffee cup. We're on the stapler, whatever. So yes, it's that the idea is that your sensory inputs are required to anchor the grid cells. The grid cells are not directly sensory driven. I say they're motor driven because here's an important thing. We'll get to this more in a bit, but when the animal moves through the environment, of course, the active grid cells change. And that can be, you could say, well, the animal's seeing new things. But even if you turn off the lights and the animal's in the dark, so there's no input that tells the animal where he is in this room. As the animal moves in the dark, the grid cells update. And so this is the idea of path integration. The animal says, well, I'm moving forward a certain amount in this direction. The grid cells sort of update their internal representation, even though there's no sensory input. And you can imagine it's a noisy process. And so after the animal moves a bit, yeah, the grid cells are updating, but you can be off. And so if all of a sudden the animal gets a sensory clue, then the grid cells re-anchor properly again. They settle in the right position again. I've talked about this. You can do this experiment yourself. You get up I've talked about this many times. You get in the middle of the night, very dark. You have to be very completely dark or put a blindfold on or something. And I come up the edge of my bed. I'm going to start moving towards the bathroom. And as you walk, there's no sensory input. As you walk, your body, you have a sense of where you are in the room. Like, oh yeah, I'm no longer at the corner of the bed. Now I'm in the middle of the room. Now I'm approaching the wall on the far side. This is path integration. And but as you approach the wall on the far side, you have a lot of uncertainty. And you're like, oh, I don't really know where I am. I know I'm getting closer, but then you stick your hand out and you start reaching around trying to feel where the wall is because you don't want to walk into it, because the path integration is not terribly accurate. It's a noisy process, and very quickly you have enough error or noise in the signal, you get a little bit worried they're going to walk into something. So it's a fun little experiment to do. And as soon as you touch the wall, then all your grid cells re-anchor again, and then you know exactly where you are. So, okay. Any questions on that?
Yeah, so with the different grid cell modules with different scale and orientation, if you kind of map it onto the brain, is it like in one direction the scale changes and then in the orthogonal direction the orientation?
No. An individual, if I recall, if Jose, if you know this, you should speak up because I know you know a lot of neuroscience. But if I recall, the orientation is also something that gets locked in by sensory input. It can vary slightly. So the two grid cell modules that might be next to each other do not always have the same relative orientation. The relative orientation can change under different environments.
I had a quick question. So in the neocortex, is anchoring kind of like identifying the pose of the object that you're looking at?
Anchoring would be, imagine you're reaching your hand into a dark box and you're feeling it. There's one of 10 objects in there and you don't know which one you're feeling, right? And you start moving your finger around on it. And at some point you go, oh, I know which one. That's the computer mouse. At that point, the grid cells in the cortex, our theory would say that's the point where they anchored and said, okay, we're going to anchor correctly for the computer mouse. And if you recognize, you feel, oh no, this is the coffee cup, they would anchor it differently for the coffee cup.
Yeah, maybe a bit like if you move on the coffee cup, as soon as you touch the handle, you know how to predict every other point on this mug.
Right.
And identify the cup and its pose.
It's interesting. One of the things I always found fascinating about this is that we'll get to this in a second, but once you recognize the object, you also have recognized your location of the object. Those are not two separate things. They have to go together. I'll explain why in a minute. But it's not like, oh, I'm on the coffee cup, but where am I? I don't know. It's more like you know where you are and what you are at the same time, because it turns out for every object you're gonna anchor these grid cell modules differently, and therefore you'll have a different location. So it's like once you know what the object is, you anchor the grid cells, and once you anchor the grid cells, you know where you are. And so it's like they're not two separate activities.
I have a question.
Yeah.
So, which relationship is fixed versus not fixed? So like, is the scale between the different grid scales fixed, or depending on situation? You said orientation is not fixed.
Right. The orientation is not fixed. The scale, I believe, is fixed. Within a grid cell module, the phase of individual cells are fixed as well. There was an experiment that proved that. So really, the thing that varies over when you're anchoring is just which of the cells that are currently active at some location in each module, and also the orientation between of that particular module to the object that's being observed.
Thank you.
Yeah. Now this is still not a very good way of determining location, because if you think about it, Take one grid cell module. Well, you've got a bunch. You still don't. If I just look at one grid cell module, it doesn't tell me where I am in the environment. It tells me I'm at one of these locations, but it doesn't tell me where I am. But if you start looking at multiple grid cell modules, then you can start really narrowing down where you are. Before I get to that, the next slide is that this. Let me show you this slide. I've shown this before. This is from this paper by this guy named Tank, and I love this image. This shows you what grid cells physically look like in the rat. I think it was a rat, and it was a mouse. I think it was a rat. And this is like a slice a slice through the entorhinal cortex. And they're showing 3 different grid cell modules. This is a physical experiment. And one module here, which has got all the colored dots in it, that's called module 1. There's another module, which is this green line. That's another module, which is this yellow line. And these are 3 modules that change in scale. He's not showing the other ones. And then what does the actual really look like in the grid cell module? It's a little bit complicated. First of all, imagine that a cell, remember, a cell can fire in multiple places in an environment, right? So you can, how do I describe this? So this little colored rhombus here, or rectangle square, is representing sort of all the places that that can be represented by a grid cell. And then, but if you keep going along the surface, it repeats, right? So a cell might be active here on this upper left dark red one, but that same cell would be active over here on the next dark red one, and the next one, and so on. So the repeating. So this is one non-repeating, and this is the one element that shows all the different sort of phases within an element that repeats. I don't know if it's confusing, but and so you can see a grid cell module actually, in this case, had 6 of those phaselets, or whatever they call them. And so what does that mean? It means these dots are actual individual cells, and they show where in space they respond. So imagine I'm a dark red cell here. Can you see my mouse? That's one cell, and it will repeat over and over again. It will repeat over and over again as you move. There's another cell over here, a different cell, but it has the same phase as this cell. There's a bunch. There's like 6. There'll be 6 in this case, roughly 6 cells that all respond in the same location in an environment. And you can imagine, as the animal is moving through the environment, there's a bump of activity in each one of these 6 phase blocks, and so it's just like there's 6 copies of the grid cells, and so as you move, those lumps, let's say the animal's moving, the lump moves across, and then it slides into the next one, and then it slides into the next, the bump of activity as the animal moves. And of course, eventually it'll run off this end, but there'll be other ones that started on the other side. So you always have 2 or 3 of these lumps of activity. Well, actually, 6 of them in this case, because there's going to be a lump in each one of these guys. So you wouldn't want to just have one sort of set of grid cells, because when you run off one end, you have to start on the other end, and cells are noisy. So there's actually like 6 copies of everything going on in one grid cell module.
So that's just redundancy? Like, what's the...
It's what's observed. It is redundancy, but it could be more than redundancy, and we'll get to that in a minute. But on this experiment, it looked redundant. Now I say that but there were some other interesting observations in this paper, which suggested it's more than redundant, and I've talked about this in the past too, so some of you will remember this. Some of these cells, can you see my mouse here this way? I'm just highlighting a cell here. And I hope you can see my mouse.
Yes, I can see it.
And it's amazing, just take this cell. And when the animal was in a particular environment, for some reason, some of these cells just would never fire. Like this particular one, not all the dark red ones, but this particular one didn't fire when you'd expect it to fire. In other environments, it did fire. And it was consistent per the environment, meaning like in this particular environment A, that cell was never, it didn't fire, but in other environments it did. And so this was very a clue to me, because it turns out if we want to have unique spaces for all the objects that we know, there aren't enough grid cell modules to do that. We'll walk through it in a moment. But we need more than 5 or 10 grid cell modules to create large spaces for all the things that a column might know. And so there needs to be another layer of encoding going on. And it almost certainly is, and we have speculations of what it is. But this was a clue because you know, this whole experiment was done on a very thin layer of cells. I think it was like, you know, 10 microns thick or something like that. So you're just getting one thin layer of cells. Of course, there's depth to this as well. And so the thought was that as you go deep into the tissue, you know, you're going to find sparser coatings of the same grid cells. And now all of a sudden you can start representing very much larger spaces. But that was speculation. But there's this one empirical clue where he did find these cells that were consistently inactive when they should have been active on particular environments. And that was like a clue that, yeah, there's some other encoding going on here that we need to, that allows the system to be more powerful. I warned you this is a really complex topic, and it is, so I'll keep going. I can go as deep as you want to know about some of these questions. Then I'm going to just pop back up here. This figure is from our paper, what we call the frameworks paper, and this is showing a couple of things. First of all, just start with the figure A. This is your classic single cell grid cell module. So we've been talking about that, and we put a little rat here to show that as the rat's moving around in this box, at different points, this cell fires. I could see it there. And then the second B here is showing 2 cells in the same grid cell modules that are just shifted by phase. So a blue cell and a brown cell, but the same basic idea we talked about before. And of course, it'd be a set of cells. And everywhere in this environment, there'd be some cells that are firing from this grid cell module. We're only showing 2. All right, so now imagine we go to C, and there's 2 grid cell modules shown here, one in the brown or red and one in the green. And these grid cell modules have different orientation and different spacing or scale. And if I were to say, okay, if I were to look at one grid cell module, it doesn't tell me too much where I am. It's some, but not too much. And the green, and if I look at the other grid cell module, it doesn't tell me too much. But if I overlay the two, and you look at cells in both, then it turns out that at any point in time, there'll be one cell in this upper grid cell module and one cell in this lower grid cell module that are active coincidentally. And that is a much more unique location than in each individual grid cell. So of course, there's always some cell in this upper grid cell module is active, and there's always some cell in this lower grid cell module is active. It's just which of those 2 cells that are active at the same time as you move about is quite unique. And if I move around in this environment, it won't repeat in the same sort of hexagonal pattern we saw here. This coincidence, these 2 cells will be occur much less often. So this is the basic idea how you can take multiple grid cell modules, and if you look at the active cells in each one, they don't tell you too much, but if you look at them together, they create a much more unique location. So now imagine if I had something like 20 or 30 grid cells, let's say 20 grid cell modules. Each one, let's say, had 30 cells that represented different locations. So the tiling was done by 30 cells in each grid cell module. Well, if you want to, what's the size of the space it could represent? It's basically 30 to the 20th power. It's like you can take one of the 30 cells in grid cell module, one grid cell module, one of the 30 cells in the other grid cell module, one of the 30, and you anchor all those at the same time. It's essentially like it's 30 to the 20th different locations you can represent, which is a pretty big number. And so if you have multiple grid cell modules, and they're all slightly different, either slightly different orientation or slightly different phase or slightly different scale, you can represent a very large space by looking at multiple cells across the grid cell modules.
So
Um,
yeah, I guess this is why I was asking like how the orientation change is like distributed over the brain area. Is like, so over how big of an area would some coincidence detector need to integrate this information, since like if the scale changes like in one direction and then orientation, I guess I'm not sure how this is distributed. Like, do we need to get long range connections to detect these coincidences?
Well, you can look at this figure here. You can see the scale bar here, 120 microns. You know, in this particular case, this is the entorhinal cortex. You'd be said, oh, if I want to go, you know, in this direction and integrate 20 different grid cell modules, which I don't think there are 20 grid cell modules, you might have to go over a couple millimeters, which is a lot. And so I don't think that's what's happening. I think that within a particular, you can almost think of this grid cell module here is, you can think of this as the base of a proto column in the entorhinal cortex. Let's see, it's like, maybe it's like a 0.5 millimeter by 0.5 millimeter or something like that. And so I think that we originally said, oh, wouldn't it be great if we could just integrate across all these grid cell modules that are scaling across the surface of the entorhinal cortex? There aren't enough grid cell modules to do that, and it's too long a distance. And so there has to be more to it. And there is. There's a lot more going on than I've just shown you here. So what I'm really showing you here is what experimentalists have shown initially, but it gets more complicated. So I actually believe that the encoding that we're talking about is when we talk about, you know, locking in on a particular part of space, it's actually not even represented by these grid cells. It's represented by something else. I can get into it in a bit. Maybe I should wait to the end because it's pretty speculative. I could talk about it now. I don't have a picture for it.
We talk about it in the end. That's fine.
Okay, just remember about it in a second. Okay. Anyway, so this is the basic idea, and we didn't come up with this idea for that you could create these large spaces, but we really liked it. So that's really cool. And we kept our fingers crossed, like, oh, hopefully we'll find enough grid cell modules that we can make this work. And the more and more the research came out, it didn't look like there were really enough grid cell modules. That, you know, there might be 5 to 8 of them or something like that. And it was like, oh shit, that's not really enough. So, but that was what was in our paper.
Quick question.
Yeah.
So about this, but isn't there actually more locations that can be encoded? Because like in A, the firing field of a single grid cell in a module, like each one of those, like the overlap in D, it's actually 2 SDRs overlapping, right? Based on the previous picture.
Well, it's not really SDRs. It's
Sorry,
two.
it's the sparse.
Really, it's 2 active cells. It's just 2 active cells. One active cell from module C, the red one, upper in C, like the upper one, and one from the red one, and one from the green one. These are just 2 individual cells that happen to be firing at the same time. And when those 2 cells fire at the same time, it's not really an SDR. It's just a coincidence of 2 cells. So we actually need a lot more than that. We need at least 20, you know, kind of active cells to detect the coincidence. You know, because we're going to have some neuron that's going to do this. Yeah.
So one of the dots here corresponds to just one of the dots on the previous picture.
Right.
Okay,
Right.
got it. Understood. Thank you.
These are the on D, we're looking at the activity of 2 individual cells. And where do those cells become active as the animal moves in this box? And the green cell becomes active in the green points, and the red cell becomes active in the red points. And in this one location, the green and the red cell are active at the same time. And so individually, the green and the red cells don't tell you too much because they're repeating over this environment. But when you look at them together, it tells you, oh, I know in this case, I know exactly where I am because that's the only place that the green and the red one are at the same time. bear in mind that at any point in the box, there will be one red cell and one green cell active. So there will always be a coincidence between one of those two cells, between one cell in grid cell module A1 and module 2. So there will always be a coincidence someplace. And so you, looking at these two grid cell modules, you could tell where you are in this box much more precisely than you could by looking at the individual modules.
So what did you
but,
mean
know,
when you said there's not enough modules? Not enough for what?
First of all, you don't want anything to rely on just two cells, right? But really what you want, if I'm going to detect a pattern among a bunch of neurons, I want at least 20 cells active, at least, so that, you know, a dendrite could, or a cell could recognize a pattern. If I want to imagine these grid cells are in the layer 6 in our cortex, and we're sending them up to layer 4. Well, I want to be able to say, oh, this sensory input is at this location on the object. Well, I need to have enough cells to do that. I can't recognize a location with just two cells. You know, cells are not always reliable. They die. The neurons aren't good at recognizing. You know, they need like 15 active synapses at once to sort of do anything. So I need a bunch of grid cell modules or something like them, so that I could recognize the location. Also, with the number of grid cell modules that we actually observe, and I'm sorry, I think it's like 8, I'm not sure. That doesn't create a very large space. Imagine now I could say, oh, it's like 30 to the 8th power. That's not that big. You know, I'd like a bigger space. I'd like the space to be unique for all objects. You know, I want to have a very unique space. I'm saying, like, I'll never get confused when I have a coincidence of cells. It's going to be like, oh, this object on this location. So you know, I really want more modules. I need more cells active to recognize it, and I need more cells to create a guarantee of have a unique location in the world.
Just to clarify, because I guess the redundancy you were showing with the tank paper would help slightly with the first problem in terms of the number of cells,
right. I'd have 6
but
cells.
it's still the issue, as you were saying, that you have to integrate over a huge area. And
right. You're good point, Niels. That's right. When we wrote this paper, we didn't know about the 6 phaselets. that was a new discovery the tank came up with. So you're right. So we actually might have 6 cells that are active in each grid cell module. But that doesn't really make the space any larger. Now, again, if I could selectively deactivate individual grid cells under different contexts, then I'd have a much larger space. You know, then it would be like, oh, you know, this cell only fires in certain locations, and, you know, anyway. So there's a lot of confusion about this, how this actually works. We made the argument in this paper that you know, a rat moving in 2 rooms, and these 2 rooms are both rectangular rooms, but one has a blue object on the wall, one has a green object on the wall, and the rat will see these as 2 different rooms. The same locations in these rooms will be encoded completely different. So here's location A, B, and C, and in a different room, you'd have a different set of cells, active D, E, and F. In fact, all the locations in this room will be completely different encodings in all locations in this room, even though the 2 rooms are the same essentially size. This also illustrates the point of path integration, meaning if an animal goes from A to C, or indirectly goes to A to B to C, it doesn't matter. Whenever you're in C, you have the same encoding for that location. It doesn't matter how you got there. Now we said, okay, this is what's going on in the cortex, but let's imagine your finger is now the mouse, and the finger's moving around in the world, just like the mouse is moving around in the boxes. And you can talk points on objects, like this point here, we'll label it X, another point Y, another point Z. That's very equivalent to A, B, and C. And here's a different object and different points, and you can path integrate, you know, as you're moving from V to T, you pass through other points like W. It doesn't matter how you move from V to T, any direction, you'll end up with the same encoding once you get up here. So that's the whole path integration component of this. Anyway, so this is the basic idea, a foundation principle of the Thousand Brains theory, is that moving our sensors through the world, a sensor patch, is very much like a rat moving around in an environment. It's the same idea, the same basic principles, the same neural mechanisms underlying it. And just like there are head direction cells that indicate which way the rat is facing in this room, we have orientation cells which represent which way the sensor patch is rotated on the object. So that was the frameworks paper, which is the first time we sort of really spelled this all out completely. Okay, I debated whether I wanted to talk about this topic because it gets really complicated, but it's a big part of the literature, so I thought I'd mention it. But if you don't follow it all, that's okay too. There's a question about how do grid cells work? How do they do this? How do they actually path integrate? How does the animal moving in the dark, why does the cell change? What's causing the cell to change to say, I'm in a new location? You know, what is doing that? It's not any kind of input from the sensors. There's something internally as the rat moves that causes grid cells to change. So this figure is trying to show how a single grid cell moving in one direction, why it would become active, then inactive, then active, then inactive. That's what it would do, right? If you're moving across an environment, like in the bottom right here, if the animal's moving, it would say the cell become active, inactive, active, inactive, active. Even if the animal was moving in a sort of strange direction, it would be like active and then active again. So we have to, there's people wonder, how could this be? And there's a number of things that point to this particular mechanism, and it's really simple, it's beautiful. I really like it. That's why I wanted to put it in here. There's basically two frequencies that are going on. There's one called the baseline frequency, which is just constantly going on. This is the theta frequency in the entorhinal cortex, I think. And it's at some like 8 hertz or 10 hertz, something like that. And then there's another frequency, that was red. Another one is the green one. And this is VCO, that stands for voltage controlled oscillator, or velocity controlled oscillator, excuse me. And this is the same frequency as the red one, but it's all, the frequency varies based on the velocity of the animal moving. So there are cells that represent how fast an animal is moving. And if you could create another oscillator that was that sped up or slowed down based on how fast the animal is moving in a particular direction, it would achieve the results we want. So these two oscillators, imagine they get added together, and so you end up with a carrier wave, this purple one at the top, where at some points they're aligned and you get more activity, and other points are just they're anti-phase and you get no activity. And so this is all, now imagine you're a single cell and you're single cell looking at these two frequencies. One, you know, they're coming in, they don't have to be on synapses even, they could be this basic frequencies in the background, but we'll come back to that in a moment maybe. But anyway, you're looking at these two frequencies. When these two frequencies are aligned, you're much more likely to become active, and when they're disaligned, you're less likely to become active. And so if the animal still is not moving, well, then the two frequencies would be exactly the same, and your activity won't change. If you're active, you'll be active. You're disactive, you're disactive. But as soon as the animal starts moving, the frequency of this green one shifts, it changes, and now your activity will change over time. And the faster the animal is going, the bigger the frequency shift on the green one, the more rapidly your activity will change from active to disactive, active to disactive. So essentially, you're taking a velocity-controlled oscillator and turning it into an on and off signal, the pace in which corresponds to how far the animal has moved in a particular direction. This is believed. There's a couple of theories about how grid cells work. This is by far the better one, and almost certainly true. There's an interesting observation that comes out of this, if you're following this, still following this. When the two have the maximum, the green and the red, have the maximum sum, where it is relative to the baseline frequency changes. You can see that right here, these vertical lines. You see this vertical line is just to the left of the peak here. Now here it's right on the peak, and now here it's to the right of the peak. This is just a natural way that this oscillatory interference works, is that where the actual peak is shifts relative to the peak of the baseline frequency. And what this means is that as the animal is moving through an environment, both for the grid cells and the place cells, you'll see that the cell starts to fire in advance of when the animal actually gets to the preferred location. And it's what they call a phase precession. It starts out on the far side of the peak, and then as you pass through it, it's right on the peak, and as you've gone beyond it, it's to the left of the peak. So there's actually additional information here about where the animal is. These prior spikes here, these prior peaks here are shifted in phase relative to the baseline. This is observed. This is a fact. So this is a very big important clue that this is what's going on inside of this. And this is a simple mechanism by which, you basically, once you can if you can get movement and turn it into a frequency shift, and you compare it to a base frequency, you can get grid cell activity. This shows one cell going in one direction only, but we can go in multiple directions. So the basic idea here is that you have to have multiple voltage control oscillators in different directions of activity. And that's what's represented by these lower ones in B here. And so a cell actually looks at not just one green voltage control oscillator, but multiple green voltage control oscillators. And if you sum all these up, you'll get the grid cell pattern. You'll get this activity of a cell. So this means that there have to be multiple, you should be able to find these cells that represent activity in different directions, and they should control voltage-controlled oscillators in different directions. And so scientists have looked for them. There's two basic hypotheses. The one I like the best is that these correspond to actually mini columns in the lower layers, that the mini columns are directionally sensitive. The cells in the mini column are directionally sensitive. And this is getting really deep here, but you need actually to get this to work for, this is just one grid cell. To get to work for all the grid cells, you actually have to have, these voltage control oscillators have to have different peaks. You have to have a set of them. You have to have a set of them in each direction. So imagine I have a mini column that's got, let's say, 10 cells in it, and each one is responding to, the whole mini column is responding to movement in one direction. And we see this is observed, right? So these cells all want to respond maximally when you're moving in some direction in space. This is in V1 and V2 and things like that. And what you need is a set of cells, each shifted in phase from each other, that are all the same voltage control oscillator. So one of the hypotheses, which we've never written down, is that within a mini column in the lower layers, you have all these cells responding to the same basically movement in the same direction, but they might be offset by phase, and which then will provide the input you need to create grid cells. So
mechanistically, it seems pretty straightforward to generalize this to three-dimensional spaces, but as I know that's not really observed, but do you have any idea why not? Because it seems like really easy to generalize that to
I agree. So that I thought the same thing. I said, okay, well, maybe the entorhinal cortex is not very good at three-dimensional spaces because it wasn't evolved for that. But in the cortex, why not, right? Why couldn't every mini column be representing a direction in three-dimensional space? And now you have a basis set of movement vectors in 3 dimensions or n dimensions, and then whatever they represent, that's what you would do, right? They would work, right? I agree. So then the question is, where do these come from? Where do what are these hard coded? These directionally sensitive, mini columns, and that's a debatable question. I like to think that they're not hard coded. I like to think that they're learned, and I've made the argument in the past that the retina has all the information you need to determine whether you're moving forward or backwards or turning left or right or going up and down. In fact, the argument, I would think the simplest explanation is that the retina sends these magnocellular cells, which are just on-off cells, and the cortex has to figure out, turn them into mini columns that represent directions of movement. So if you ran these magnocellular on-off cells through a spatial pooler, you would end up with multidimensional representations of direction. That is, whatever the data contains coming from the retina, if it's three-dimensional, it would represent 3 dimensions. If it was 2 dimensions, it would represent 2 dimensions. A spatial pooler would do that. It seems on. So if we assume the system learns and the information is coming in the way we think it learns, I don't see why it wouldn't represent 3 dimensions. Or if the data was n-dimensional, it would represent n dimensions, but we typically don't get that from our retina. If some of you might know, in the grid cell literature itself, so now we're going back to the entorhinal cortex, they've done a whole bunch of experiments to see can grid cells represent 3-dimensional space. So they've done experiments where rats are climbing around on lattices in 3 dimensions. That wasn't very definitive. Then they did experiments with bats. And these are amazing experiments. They have the bats flying through these very long, large tunnels. I mean, like, you know, like a quarter mile long. And they're recording from the cells in the rat's entorhinal cortex as it's flying through this space. It's amazing. And they can see where in the 3-dimensional space of the tunnel or the environment that the rat is. And they say, okay, well, do the grid cells represent 3-dimensional space or not? And what they found was kind of a little disappointing. They found that the grid cells work the way you would expect them to work based on landmarks on the ground. So like the grid cells represent a nice 2-dimensional map of the surface of the Earth in some sense, or that the rat is flying. The grid cells do become active in vertical dimensions as well, but they lose all of their normal grittiness. They no longer act in some sort of regular way. Like a cell that becomes active at some point up in space is no longer has the same scale and, you know, phase and orientation. It's just like it's wonky. So the grid cells, they still become active in 3 dimensions, but they just the third dimension just messes them all up and doesn't look right. So no one knows what to make of that. So, but I still like to believe that in the cortex, it can be 3 dimensions, and I don't see any problem with it in terms of Monty. Even if it's not that way in the brain, I don't see why Monty has to, you know, stick to this historical artifact of 2D. All right, so that's oscillatory interference. There is another theory called continuous attractor networks. And just in case you know that, it tries to recreate the same basic behavior of good cells, but it takes far too many neurons to do it. There's not enough neurons to make it work. But it's complicated. But someone made an argument. There's a paper where they argue that both are that both oscillatory interference and continuous attractor networks are both active, and I think that was the right argument. The reason I say this is because if you look at this system right here, when the animal stops moving, what actually happens is these voltage control oscillators stop firing altogether. And so there would be no persistence of memory as to where the animal is. That is, this works for path integration as the animal's moving, but when the animal stops moving, these signals disappear, and then you might not remember where you are. And so you need an attractor network to sort of lock in cell activity even when the animal's not moving. And so like, okay, well, we'll stop here and keep everybody active. And then we'll continue moving again in the future. So the argument was made by one researcher that the combination of continuous attractor networks for permanence and oscillatory interference for path integration is the right answer. And I think that's probably right. It's all complicated. So now I've gone through most of my stuff. Yeah, go ahead. Your question?
Yeah, probably just wild speculation, asking you to wildly speculate. So because abstract knowledge, right? So looking at this representation here, it seems like you can create a grid in n-dimensional, like you were saying, and is that a path to representing any abstract knowledge spaces?
Right. Yes, the answer is yes. And there's a fair amount of research on this as well. First of all, the entorhinal cortex is known to represent all kinds of things, not just not just where you are, but it seems to find a general map. You know, remember the hippocampal complex, which includes the hippocampus and the entorhinal cortex, is where we store our short-term memories. You know, things like, you know, your episodic memories too. And it's not just like where you are. It's like, what did you do? What did you have for breakfast? And so on. So all these things are stored in these same cells. In fact, there's all this research that says that the grid cells and the place cells somehow are able to encode all kinds of stuff. Yeah, and even the same exact cells are able to encode different types of things. So the basic belief is that this representation of space is underlying all kinds of both time and space and all kinds of things you might call abstract type of stuff. I don't know the current research on that. It's a little confusing, but this is clearly happening. I've speculated that, you know, in the cortex, you know, if we're trying to understand abstract spaces, the only thing we have to explain is what is the equivalent of the voltage controlled oscillator? Like, what is movement when you're thinking about politics? You know, what is movement when you're thinking about mathematics? You're not physically moving something, but you need to store information at different locations, and therefore you have to do some sort of path integration or some sort of, you know, and it's very confusing. I've argued, and I wrote in my latest book that like you might think of like a mathematical operator as a type of movement. You know, you might say, well, when I do some transform or I do an add property or something like that, that's kind of like a movement. And if I and therefore by performing some operation, you're actually moving to a different space and you're starting the locations in the different spaces. It's not clear at all exactly how this occurs, but it is clear that this mechanism is underlying it all. I mean, I think that's very extremely likely that the same mechanism is underlying all kinds of our thinking processes. All of them actually. So it's a bit of a puzzle. Like, oh, exactly how does it do that? I don't know, but it's doing it. It's clear that it's doing it. It's not. There's no other mechanisms anywhere. You know, you look at cortex as cortex. It doesn't look like it's going to be different anywhere else. And we know the entorhinal cortex does multiple things, so it clearly can.
It almost seems like we, even with abstract concepts and spaces, we kind of try to map them onto 2 or 3-dimensional space. Like, even when we think about math, we usually try to visualize it and plot it somehow,
Right.
like in
Right.
3D. Like, it's really hard to think about concepts that are in higher than 3-dimensional spaces. And even when we talk, we usually use like metaphors that talk in terms of spatial navigation and movement.
Right. It's clear the brain wants to drop things into this mechanism because that's all it has. And everything humans do, it works to some extent. You know, it's interesting to speculate. You think about like the transformer networks and the large language models, and they're so good at language. You know, they're better than humans in many ways. Some ways they're worse, but better than humans in many ways. And why would that be? It might be because language is somewhat is a poor fit for this mechanism. You know, this is the mechanism that evolved to explore environments and objects and so on, and we're shoehorning, you know, language onto it, and we're proud of it as humans. But you know, maybe the reason that ChatGPT or these large language models are better at just modeling language because that's what they're designed for. They would do terrible at this kind of stuff, you know, recognizing, you know, feel of a coffee cup. They can't do that at all. But you know, maybe if you design an algorithm that's just for language, it can be better than us because we're shoehorning language into this mechanism, you know, and that's why math is hard. There's a lot, you know, calculators have no trouble with math. We do. So it's because we're fitting all the stuff in. Now, this is not bad news for the Thousand Brains project, because in the bottom line, we're going to want to create intelligent machines that interact with the world. And if you're going to create intelligent machines that interact with the world, well, this is the way to do it. You don't want to use a large language, you know, you don't want to use transformers. So our project, the Thousand Brains project, is really biased towards robotics and physical manipulation of the world, and can do other things too. but it may never equal, the language abilities of ChatGPT. But it'll understand what a cat is and what it feels like and how it sounds and it will have real knowledge of the world as opposed to just knowledge through the correlations in language. Okay, so let's see how we did on my, it took a long time here. I just want to point out a few things here. I'm in the unknown section here. We just talked about, are they 2D, 3D, or ND? We just talked about that. I'd like to point out that in the neocortex, in the entorhinal cortex, you don't find just grid cells and place cells and head direction cells. You find the majority of cells are combinations of these things. I think the word is conjunctive. I have to look that up. I think that was the word for it. Most cells are somewhat like grid cells and also somewhat like head direction cells and other things. There's this huge mixture of in-between stuff. And there's other cells that are like, instead of just being place cells, they're like object-oriented, object vector cells and border cells, and these cells that seem to respond to particular things in environments. So it's a messy mix of stuff. And the scientists they look for the grittiest grid cells when they want to do grid cell research, because the other ones are kind of half grid cells and half direction cells, and it's a mess. So I just want to, but that's a misleading picture I just showed you. It's like all these other cells that are in between these things. I also talked about in the tank paper how sometimes grid cells don't behave the way they're supposed to behave. There could be a lot of those. And then we've already touched on this, but the place cell and the grid cell interaction in the hippocampal complex is very much analogous to the layer 4 to layer 6A interaction in 1,000 brains theory. And we speculated that layer 6B is orientation, and that would be like head direction cells in the entorhinal, in the hippocampal complex. So this is a very strong analogy. It seems it's almost certainly correct. And that's how we do this all the same. So that's all I was going to present about grid cells at the end of my
Quick
slides.
question for the conjunctive cells. Is that, like you said, there's a mess there, some of them do a little bit of bunch of things. Is that a biological constraint? Like, do they grow like that? Or is that like exposure to environment and they settle into those patterns?
I don't know. I assume it's part of the mechanism. So let me just, I talked about how grid cells on their own are not really sufficient for representing the spaces we need. They're just not enough grid cell modules. And if I think a grid cell module is really just one grid cell module is equivalent to what you get in one cortical column. There's just not enough grid cells to make the space. So, but we do know that there are there has to be cells that are voltage, directionally sensitive, phase shifted cells. We've observed these conjunctive cells, which are combinations of grittiness and direction. And it seems to me clear that when we actually recognize a location in space, we're not really just looking at the grid cells. We're looking at all these other cells. That is, layer 4 is not just looking at the output of the equivalent of grid cells in layer 6. It's looking at the equivalent of all these cells in layer 6. There's a lot of other cells down there that are going to be doing these weird things. And somehow the combination of them is unique. And I don't have a better answer to that right now. So I think it's more that it's part of the mechanism. It's not, I mean, yeah, what they represent is going to be learned, but the fact that there's direction cells and grid cells and there's combinations of them, I think is going to be part of the evolutionary mechanism. It's not, and then.
I don't know if yeah, and that was a good segue, but like you mentioned earlier that at the end you could talk a bit more about how you feel like, yeah, encoding of unique locations might actually happen in cortex, like given the limitations about number of cells and things like that.
Right. Well, again, go back to, let's see here. I don't have a good picture of it, but remember I said these could be like cells in individual mini columns, and they're directly sensitive. And then I said that if in the mini column you have to have multiple cells at different phase shifts. if I'm going to get a complete grid cell module, I have to have cells at all different phases of the theta cycle in a mini column. So I might have 10 cells or 20 cells that are all firing at the same voltage controlled, velocity controlled oscillator frequency, but they're going to be shifted from each other. So if I looked at those cells, again, I'm looking at all the different cells in the mini columns, and the mini columns are representing different directions of motion. A grid cell would, in some sense, if I could look at all the activity across say some of these mini columns are active and some of them aren't, and I look at all the cells in those mini columns and they're all phase shifted, but they're all, I would have a unique coding for location. I'd have plenty. I'd have, for example, even if I assumed my mini column is a 0.5 millimeter by 0.5 millimeter, I might have 100 to 120 mini columns. Each mini column may have 20 cells, and so maybe a third of those mini columns would be active. So I might have, like, say 40 and times 20. I might have 800 active cells at that point in time that are representing that I could encode a unique location for. That is, if I looked at which cells are in phase, aligned in phase right now in these different mini columns, that would be that's the information I need to update my. That would be all the information I need to know where I am in this object, in which direction I'm moving. It would encode both of those 80 cells or whatever I came up with, the 40 times 20, 800 cells. Those cells, if I looked at which ones are in phase at any point in time, they would uniquely code a space and a direction, a highly unique space, a location in a particular object in a particular direction of movement. So the information is encoded there. It's not in the form I want it, but it's there. I got a lot of, and all those mini columns can anchor differently. That is, which cell is currently in phase with the baseline frequency. That is the anchoring process. So I basically, I have 140 active mini columns and I get to pick which of the 20 cells are in phase at this moment with the baseline. That's 20 to the 40th power of unique codings at that point in time. So the information is there. How it gets extracted, the problem, as I said earlier, one of the problems is that when you stop moving, well, that information would be unique plus direction. So I don't want to just, I want to have a way of not I don't always want it to be direction sensitive. Anyway, I'm talking sort of sloppily here, but if we wanted to, we could have a little brainstorming session. We could try to walk through exactly what would be required to get the information we need. But my point is, there is enough information in these cells to encode exactly where I am on a particular object with a high space, with very high dimensional spaces. And in these cells in these mini columns, which are required to generate grid cells, would have the information required. Again, it may not be in the form we want it, but it's there. So that to me is like, okay, we're not combining all these grid cell modules. We're actually combining the information across multiple mini columns. And there's 120, a minimum of like 100 mini columns in a small cortical column. So that's enough.
Yeah, I think that would be interesting to talk a bit more in detail at some point. Because I'd also be curious to better understand like how we could do path integration in these like n dimensions without having to create the grid cells, like the n-dimensional grid cell or whatever.
I think
just because like if these are kind of unidirectional, then like even just in the simple case you move like right and then move back, like the same cell won't be active.
You won't have that's why I said the information is there, but it's not in the form you want it, because those mini columns would be directionally sensitive. So the hand wavy argument I have here, Niels, is the following. To me, what we actually see as grid cells is the output of the continuous attractive network. It's the memory of where we are at some point in time that persists even when I stop moving. but the actual encoding of space is not in the grid cells. And so somehow if I move in one direction, I turn and move back in another direction, well, I have a completely different set of cells that are active in the mini columns because different sets of mini columns are active. And so what's required is that somehow we anchor the second set of the new reverse set of mini columns. We have to anchor them appropriately. And so I have to have memory of what that should have been, and somehow in some hand wavy form, I would say the grid cells are somehow saying, okay, we're here right now. We're going to lock down. Now, if I start moving in another direction, I have to reinstate the active cells in the mini columns in their basic phases. And so it's sort of like, yes, it's a combination of these two things, sort of like a sort of a permanent memory, or memory that stays somehow active that allows me to reinstate the phases that I need for path integration. So it's a combination of this oscillatory interference, which is very specific, directional specific, and the grid cells, which are more like a continuous attractive network type of behavior. But this is all very hand wavy. But what I feel good about it, Niels, is that I think the information is there. It's not like,
Right.
if I couldn't figure out how this possibly could be represented, I'd be nervous about it. But it's like, oh no, it's there, and these things are all observed, and this phase precession is observed, and these voltage controlled oscillators are observed. And grids, they're there. It must happen inside of a cortical column. So the information is there somehow. And even if we don't understand all the details yet, I'm sure it's understandable somehow.
Cool, thanks.
I have a question about velocity. Like, velocity itself is not that's a calculated quantity. It's not a primary signal. Like if you're moving in the dark, you can't see or feel how fast you're moving, but you know.
Right.
So how does
Right.
that come about?
Well, I don't know the answer to that question. I would suspect initially, this might have been, hardwired from an animal, like a rat moving in an environment. It could be a hard-coded somehow. It's subcortical. It's clearly there. I mean, I don't know how the neurons calculate it, but when the animal starts walking, these things change. So there has to be some way of doing that that doesn't rely on sensory input. I'm not worried about that because that could be a very hard-coded evolutionary thing. When
So
it
it's
comes
just calculated from motor.
I don't know,
motor
some motor
neurons.
system, right? Subcortical motor system. I don't know how it does it.
I think some
when it-
people. I think in the MEC, I think besides grid cells, place cells, head direction cells, like some people say there's also like speed cells, and maybe it might be related to that.
there are, that's what it is. But
Yeah.
so they're there. The question is, I think the question was, well, how do those speed cells how do they know how fast you're moving?
That's a
Right?
research question for somebody.
Right. But my argument is it's not a generic algorithm. It's probably a hard-coded thing that animals had to evolve at some point,
Yeah,
you can
makes
imagine
sense.
like a little animal that's just walking in one direction. It's somehow there's a signal that says move your legs, and that move them faster, move them slower, and then that signal becomes the voltage. It becomes, I don't know. It doesn't bother me.
Also, for like machine dead reckoning, you typically use gyroscopes, which are just internal only. And so for human equivalent could be vestibular system as well. So as you're moving forward,
that's a good point.
gives you like
Right.
an internal gyroscope.
Right, exactly. I reminded my vestibular system after I had my bike accident because it got messed up, and boy, did I have some bad vertigo.
Yeah, but that's a accelerometer, not a velocimeter, right?
Well,
If you're
yeah, but
moving
you
at a
could
constant speed.
Well, you
then
could use
yeah.
that to set a constant speed and then change it. Who knows?
It's like it must be learned, at least in some cases, like when you start driving a car, you kind of have a model of velocity there, even though we couldn't possibly have evolved that.
Right.
Yeah, good point.
Right. And I mean, you could fool people, talk about you only detect acceleration. with your vestibular system. But yeah, it's very easy to fool people. You can, in these theaters that have like 3-dimensional, like these moving seats and so on or whatever, you can accelerate someone quickly and then slow it down, and they still perceive that they're moving. You know what I'm saying? You can fool the body into thinking you're still moving based on just acceleration. So anyway, there's a lot of ways to do it. I do think it's interesting. Again, I love the fact thinking about the retina, because it's clear that we get all this information from the retina. That again, if you imagine you're just watching someone playing a first-person video game, and you know how that person is moving. You know if they're moving forward, backwards, jumping, turning their head, ducking. You know all this stuff, and it's all coming from the visual signal. And if they're walking forward, you know that. Your grid cells are updating as you're doing that, even though you're not controlling anything. So it's all coming from the visual signal. And as far as we know, the visual signal from the retina is just these on-off cells that, and so it's able to say, like, it's able to take patterns that are changing on the retina and seeing which ones are turning on and off and their relative distribution across the space of the retina, and turn that into a very clear signal. You're moving forward, you're moving backward, you're turning left, you're turning right, you're looking up and down. It's there. So all that information has to be decoded from these on-off signals, and almost certainly it's learned in the cortex. So I just think it's another place like, wow, isn't that interesting that you can determine your movement just from these bits turning on and off in your visual space. It's all very clear.
Yeah, I guess I think of movement as like not just the grid cells and the place cells, but all other information, sensory information that we're getting, like what Tristan mentioned about vestibular system, and what you just mentioned. I guess it's kind of optical flow
Right.
information, and even I guess proprioception, and even like signals that we send to our muscles can probably tell us something about. I'm not sure, actually. Maybe not that
I'm looking
one.
up the. I want to go to the our paper.
Just a note on the vestibular system. It is awful at estimation, by the way. Like the first thing you learn for flying is to not rely on your vestibular system and to just ignore it completely, because it
Is
will.
that right?
Yes. And so like if you truly isolate it and just have your vestibular system, it is terrible at figuring out what's actually going on.
I'm trying to find our paper that we've been working on. I haven't looked at it in a while.
I think if you go on TBT paper drafts.
Oh, here we
the
go.
first
Right. And I want to show you this this figure. And it's compiling it.
But it looked like you had it open as a PDF too,
Oh,
actually.
did I looked for it. I didn't see it.
TBT paper drafts 15.
Oh, right there. Oh, you're so much more observant than I am. Thank you. What would I do without you guys? I'm going to show you this figure in here. Come on, fasten that. Well, did I do it? Is it? Is that no? Keep going here. Here, this figure. And this, and then zoom this in. You guys can see this, right?
Yeah, thank you.
So this is the figure from this paper, and notice here we're showing this is like a V1 column, and we're showing two inputs to it. One is the green one is like the sensory feature, and the other is the sort of motor signal, which is updating the grid cells, if you will. And I show there's multiple sources for that, right? One is I show from the retina itself, this large red arc suggests that these are the magnocellular cells that are detecting motion on the retina, which is sort of ground truth. So the eye movement ground truth from the magnocellular cells. But we also know that projecting to the exact same cells in the thalamus. So this goes through the thalamus when we're arguing that the thalamus changes the orientation of these signals. The exact same signal. There's the superior colliculus projects to the same cells that the magnocellular cells. So the relay cells in the thalamus get input from. They get input from the retina, but they also get input from the superior colliculus. They also get input from the vestibular system. These are known connections. So in some sense, all these are valid sources of movement data. The vestibular system does project to LGN in the thalamus, and it would say, hey, given your head movement, I think, you know, you're changing the orientation of your head. And this is what messed up when I had my accident. I messed up my vestibular system. This happens to anyone gets dizzy. And so the vestibular system is sending incorrect signals saying the world that you're turning your head when you're not. And then the thalamus tries to correct that orientation so that it starts to rotate the signal, and it looks like the world is rotating because the vestibular system says your head is moving. And so the world starts spinning around because the thalamus is trying to compensate for the head movement, which isn't occurring. And my point is, there's multiple sources. There's many different sources that can provide the movement data. And if you just look at what the inputs to the relay cells are in the thalamus, you can see the different sources.
Also the motor efference copy, like from the motor output.
That's from the suprachiasmatic. Also this one.
From the 5, yeah.
Right. I don't know if but that doesn't affect column 1, right? That would affect
Yeah, to column 2, I mean.
Right. So there you go. On this signal right here, this is the same. Oh no, wait a second. This one here, it comes out, and it then becomes also an input to these relay cells in the next ones, right? So that's the other input. So this column can say, oh, I'm telling you to move the eyes, and it says, okay, well, I'll take that information and I'll assume you're moving the eyes. Anyway, the point is there's lots of sources of movement data, and when it comes to the Thousand Brains project, we have to be quite open to new ideas and things we can make up and stuff we could do. We don't have to stick to any of this. If we can make up all the stuff we want to make up.
Yeah, there's also direction selective retinal ganglion cells that project to the shell LGN, which project to layer 2-3.
Is that right? You know, I had read all these years that all the cells from the from the retina were center surround cells, but in rodents,
Yeah,
they're directionally selective.
yeah. Well, they have both. They have both types.
Who does? The rodents?
Rodents do.
Do humans have any directionally sensitive cells? I'm not aware of that.
That's a good question. I don't know if they have been able to investigate that or not.
Well, it's not just humans. It's also monkeys, primates. So this is a very interesting point, because you would think, well, why wouldn't the retina just tell you the direction it's moving? And therefore, the cells coming off the back of the retina would be directionally sensitive cells. And we see that in simple mammals like rats and mice. But we don't see directionally, as far as I know, they don't see directionally sensitive cells coming off the primate retina, which tells you, this is really cool, because it gets back to how many dimensions we can deal with. It says in us and other primates, actually determining which direction the eye is moving is not determined. The eye doesn't tell you that. It has to be calculated in the cortex itself, because the cortex is not, as far as the ganglion cells in the retina and the relay cells in the thalamus are not directionally sensitive. But as soon as you get to the cortex, they are directionally sensitive. So it tells you that it's taking this raw information and then determining what are the possible movement vectors. Whereas in the rat, it's more hard-coded. It's more like, oh, I'm going to tell you what the movement vectors are. These are the movements you can make.
You know, it could be related to the fact that rodents rely so much more on the superior colliculus, that there has to be, it's more beneficial to them to have performed some of this direction selective stuff earlier in the pathway.
Yeah, and they also have the kind of shallower visual hierarchy.
Right.
So it's just kind of like compressing everything.
Again, it's, we go from this idea that this whole system started with navigation on surface of either the bottom of the ocean or the land, and it was a two-dimensional thing. It's really specifically designed for getting to your shelter, finding food, getting back, and then over time it got more and more general purpose. And this is one of those things that makes it more general purpose. In the humans, we don't assume that we don't make any assumptions about what that motion actually is. But this gets back to the interesting about the kind of idea of abstract thinking. what we're seeing here in primates is that there's a more primitive signal, this center surround signal, that again gets decoded in the cortex into like, okay, what are the directions is this thing actually moving? And one could imagine, what if you lived in a 4-dimensional world and the retina viewed it? Would the column learn to represent those four dimensions? Because it makes no assumptions at all about the space. It just says, I'm going to start with a bunch of on-off, center surround bits, and we will deduce from that what are the different directions we're moving. Yeah. So anyway, I think we see this evolution from like the simple animals to more sophisticated mammals, where you get more general purpose principles going on. Pretty fascinating. I think so.
Yeah.
I think all of these kinds of presentations always like at the end makes me appreciate like, wow, like the cortex is amazing. Like getting all this information and making sense of it. And it's like,
Right. And it does it all within a single column. I mean, it's,
yeah.
that's amazing. Yeah, it is pretty incredible. So I love that constraint, Jose. It's like, okay, it's gonna have to happen. It's gonna be there. It's there. We can find it. It's these things all exist. Yeah. Again, I want to stress one more time. We want to understand this stuff, but then we have to build systems that work on whatever principles we can build them are more practical, and we don't have to emulate all this stuff, which I think is everyone understands that pretty well. But it's worth staying over and over again. I don't want anyone to say, "Oh my God, it's so complicated. How are we ever going to do this?" Well, we don't have to do all this. This is the way the neurons do it.
Yeah, thanks a lot for presenting this. This was a great discussion, and yeah, just a really nice overview.
Okay, I was worried it might be a little redundant for some people, but maybe not. I don't know.
Yeah, it was
Yeah, I
really
feel
nice.
like every time we revisit this stuff, I learn something new. So
All right.
yeah, it's
All right. Well, it was fun doing.